and we will talk about the agenda first. As always, if you have questions as we're speaking, you can type them in the chat or you can write them down and unmute yourself and raise your hand at the end when we do a Q&A. We love taking your questions and chatting with you all at the end. So our agenda for tonight. So Yazie's going to start with a little bit about why we're here and what we've learned through this webinar process so far. Then I'm going to give a very brief version of our EnSoil 101 talk, which we will be having in 3 weeks with Meredith. So if you have not joined us for one of those EnSoil 101 webinars, please come. They're awesome, and we go a lot more in depth about the mechanism of the algae. I'll also talk a little bit about soil testing tonight. Soil testing has really influenced Russell's work, so we just want to give a little preface for that. Then we're going to give the floor to Russell and talk about his corn. Then we'll take your questions. And as always, in the end, we encourage you to share this knowledge with your local communities, because that's what we are here to do. So brief introductions. My name is Camille. I'm the Director of Partner Education at Enlightened Soil Corp. I am also a regenerative livestock farmer up in Michigan, and I'm out there working with the animals and growing grass and really focusing on soil health. So this work means a lot to me. Yazzie?
And I'm Yazzie. I'm the Partner Program Coordinator with Enlightened Soil Corp. Also run the Instagram and the socials and was promoting this. And I live vicariously through Camille every day with her, all the animals she's with.
Awesome. And we'll have Russell introduce himself when he comes on here in a bit.
Okay, so I just wanted to start by talking a little bit about why we felt the need for this webinar and what it means to us. So one of the biggest lessons I'd say we've learned from working in the agriculture industry is that it takes way more than just having a good product to gain farmers' trust, as it should. For anyone that isn't a farmer or doesn't know a farmer, farmers work so hard. I could not have imagined the blood, sweat, and tears that go into farming, and frankly, don't get enough in return sometimes. But thankfully for us, they're passionate enough about the land and the food in their community to keep going. And with that being said, EnSoil Algae, it's a powerful tool, but it's only as powerful as the community we build around it and the trust we build around it. So that's our goal here with the webinar. We just want to create and nurture a space where people can come and learn about the algae as a tool, but also all the other tools come with, questions and laughs and just work together to be successful as a whole. And in fact, just about community and how important it is, we came to know Russell and his partner, Liz Haney, by attending their community building events, specifically the Healthy Soil or the Big Soil Health event and Beyond the Yield. Their community of farmers and them have been such a catalyst for our growth. We're just really appreciative and want to keep spreading the love. And we're really excited to see Russell and Liz Haney at this year's Big Soil Health event in December in Iowa. And hopefully we'll see some of you there. But yeah, thank you all for joining and being part of this community with us, and ask questions and just feel comfortable in the space to be curious.
Thanks, Yaz. You want to talk about some of our upcoming events?
Oh yeah, so speaking of community, we're going to be attending the Sustainable Agriculture Conference, November 11 to 13. I went last year. It was the first time I met Russell, actually, in his big black cowboy hat. And it's a really fun event. If you live in the area, I'd go. And then we have our next EnSoil 101 webinar with Meredith, who is just amazing. You can scan that QR code to sign up in advance, but you'll get a million emails from me about it too. And then the Big Soil Health event, seriously not one to miss, and that's in Iowa, and our team will be there, and we hope to see you there. Over to you, Camille.
Great. So let's just talk a little bit about what is EnSoil? If this is your first webinar joining us, welcome, and we are super glad you're here. So we grow a live strain of green freshwater algae. The strain is called Chlorella vulgaris. And Chlorella vulgaris is a really powerful biostimulant in the soil, which means that it encourages the natural processes of nutrient cycling and soil microbial activity to help stimulate and accelerate growth of plants. So we are seeing this be super effective with crops of all different kinds and in all different geographical locations. This strain of algae is actually present in the natural ecosystem and in all ecosystems around the world. So it's not an invasive or anything like that. And we are seeing 4 main mechanisms for the way that the algae is working with the plants and with the soil. So at our EnSoil 101 webinars, we go through this in a little bit more depth, but tonight I'm just going to run through them so that we have a background for Russell's talk. So first, the algae is stimulating and growing the microbial populations and the diversity of soil microbes under the ground. And so this allows the plant to, well, microbes are cycling nutrients. That's their natural job in the soil. And this allows plants to take up those nutrients and therefore have their growth stimulated. These are things like carbon and nitrogen and phosphorus in the soil. Additionally, EnSoil algae enhances the chlorophyll. Chlorophyll is the green pigment that you see in plants. So it enhances the chlorophyll and it improves the photosynthetic capacity. So it allows the plant to do gas exchange, draw down carbon, absorb more sunlight as energy, and improve the process of growth. And then finally, the plant's tolerance to stress is improved through the mechanism of the algae. And with that, we're seeing an increase in water retention capacity, specifically in areas where there are droughts, which I know a lot of you are in that space right now. And then we're seeing more water infiltration. So the structure of the soil is actually improving with the EnSoil algae. So if you want to learn more about these, we have lots of resources on our website. Please reach out to any of us. We would love to chat with you more in depth about the mechanisms. And then please come to the next EnSoil 101 webinar where we talk about these more in depth. So before Russell talks, we just want to give a brief history of soil testing and talk a little bit about the Haney test, which has been important in Russell's work. So soil testing in the US has sort of started with these fertilizer companies forming and measuring nutrients needed in really what we call dirt, which is non-living and non-dynamic soils. So dirt that does not have a community of microbes and is not really cycling nutrients in an appropriate way. Whereas soil is something that's living and dynamic. It's constantly changing. It has these habitats and structures that tons of different microbes, bacteria, fungi, protozoa, nematodes, they all live in. And so when the soil testing started with fertilizer companies, they were measuring nutrients that were needed in the non-living soils. And they didn't really evolve these tests to measure for any microbe activity or nutrient cycling done by these microbes. So specifically, this resulted in a large over-application of synthetic fertilizers. And this obviously cost the farmers more money, but also put a lot more chemicals in our watersheds. So drinking water became heavily polluted for animals and humans. And we just saw way too many chemicals be put in the ground because of these recommended nutrients from the soil test. So how this differs from the Haney test is that the Haney test is a soil test measuring the biological activity. So it's actually testing for the activity of those microbes. It's testing for the nutrient cycling. And it is providing nutrient recommendations. Sometimes it will provide cover crop recommendations or percentages of grasses versus legumes that would benefit the area that's been tested. And then it provides a soil health score. So that is sort of a conglomeration of lots of different measurements. And we're not going to go deep into all the things that the Haney test takes tonight. But again, if you are interested in that, please reach out to me and I'm more than happy to talk with you about it and share more resources. Some of the things that are relevant to Russell's work tonight are what we would call the food and the energy sources available to the microbes. And these acronyms here that you see stand for water extractable organic nitrogen and water extractable organic carbon. And sometimes these are pushed together as water extractable organic matter. But really, this is the food source and the energy source that's available to microbes. The food comes from nitrogen, which is made in amino acids are made of nitrogen, and amino acids make up proteins. And then carbon is this energy source for the microbes. So the Haney test looks at these values. It also looks at the carbon to nitrogen ratio. It looks at the soil respiration, so what's going on with the gas exchange in these living communities, and also the soil organic matter. And my metaphor for this is sort of like the house or the habitat under the soil. So we see increased soil organic matter often with the use of EnSoil. And this is why we get things like improved water retention, because the structure of this habitat is stronger, and therefore it can be more porous and more water can be retained in those spaces. So this is just a little window into what the Haney test can measure. It's a really powerful tool, and we recommend it for all growers of all different kinds. So please reach out to us if you're interested in more information about that, and we'll definitely answer any questions you have about that after Russell talks. So now
I do have an important question for Russell too.
Yep, please, Yazzie.
Russell, we're all very curious. Oh, can you talk? Okay. Oh, we're all very curious. Oh, can you? I just saw your message back in two.
Yeah.
Okay, there. We're all very curious. How do you go about selecting your music for your TikToks, for your farming TikToks?
I would say it depends on how many beers or bourbons or how tired I
am. Thank you. Just kidding, kind of, but anyway, introduce yourself.
Can you hear the beeping?
Nope.
Okay. So I'm Russell Hedrick. I'm a first generation farmer. We farm predominantly corn and soybeans, sometimes small grain, and we tried to learn how to use regenerative practices in a systems approach with soil testing and biological testing, and really anything. I would say our farm is more of a research farm than it is a normal grain farm. We test an absolute ton of products, and there's been a lot that we've tested that really haven't met the mark, and there's been a few that have really done quite well. One of the reasons I'm running a little bit behind tonight is the only downside I can tell Yaz that I've seen with EnSoil Algae so far is my soybeans are so big that they actually would not feed through the auger of the combine without me jumping in there and actually making them go through. We're picking our yield plot this year, and we started about 10 o'clock this morning, and we still have to go weigh. But I mean, that's the only problem I've seen with EnSoil Algae is I've had to get in the grain tank and have to force the grain out of the combine. So I'd say it's going pretty good for them.
Yep, I'd say so.
But we still use fertility. We still use a small amount of chemistry. I mean, on average, we've reduced our chemistry at a minimum 80, sometimes 90%. Fertilizer, I would say that we're using at a minimum 50 to 60% less than our neighbors, and looking at ways that we can actually do testing like they were talking about, the Haney test. We've used that, the PLFA. This year we have some results from a new soil sampling company here in my state from Rise Bio, looking at the metagenomics, and we'll talk about that a little bit. That was pretty cool. Looking for ways to farm and be profitable. At the end of the day, doesn't matter how big you are, if it's a half acre or 1,000 acres, we're all working our tails off, and we should all be able to have a comfortable living. And so that's why we look at products, and we've got 2 years of data now from EnSoil Algae on corn specifically. It's been our biggest achievement. looking at return on investment. So I would say, my recommendation is even if you do it like we do and start on some smaller acreage, try it out, see what it's doing, and do the testing to verify and be able to see what you're seeing. Because as many farmers as we've worked with, they talked about our network earlier. We've seen tons of farmers see the benefit of this so far. And so what you're looking at here on the screen was last season. We did some trials where we ran it in furrow, and then we came back and we tried some different application timings on the foliar spraying. And the picture on the left was the untreated plot, and the picture in the middle you can see even having the InSoil in furrow 30 inches apart, we could tell right down to the row as far as plant vigor, seedling emergence. We saw some really good uptick in those numbers. One of the issues that we had is we typically plant corn and we know how many weeks we have until we can go out and post spray it. The only issue we had last year is our corn grew so daggone fast that we had to switch up some of our spray practices for our foliars and had to have it grown, do a little bit extra work just because the corn grew at such an exponential rate. And you can see, on the right hand of the screen there, that was in the plot where we had treated with the algae. And, we're talking about less than, 3 feet apart, and you can tell a difference in the corn plant. And that's just absolutely astounding to see, that much plant growth different. I mean, we were talking 2 leaves. So when you look at, maximizing sunlight inception and the liquid carbon pathway that, C3 and C4 plants are putting that liquid carbon into the system to feed biology, it's kind of like a double whammy. We fed them in the beginning, and then we also had, something season long that was performing better as far as the plant that was feeding the biology even more. You want to go to the next slide? And so, this is what we did with Rise Bio. These are some of the metagenomics. You can zoom in on the screen, but we've got observed genre, mycorrhizal. We saw an uptick with our mycorrhizal, our fungal to bacteria ratio, which is what we talk about a lot in nitrogen management. We want to be balanced, especially in our soils in the Southeast where we have a low CEC. Having a higher fungal to bacteria ratio where we're more balanced. We don't see nitrogen loss near as much. And it's a way for us to maintain that nitrogen in the soil. Nitrification was higher and denitrification was lower, which is what we would expect. So, it's like I talked about, we're using these products and try them on the farm, but, scientifically, there are ways that we can verify what we're seeing from these products as well. And it's really shined through with all of the 3 tests that we really put it to the measure with. Like, yeah, do you have anything you want to add, or Josh, would you want to speak on this for just a minute? Is that okay, Yaz?
Yeah, absolutely. Hey, y'all. Josh Cole here with Rise Bio. So first thing, I think, Russell, your soil was incredibly healthy, and so off the bat, we saw that Russell's soil is really healthy. One thing that the biggest spike that we saw was in observed genera, and that's just the number of microbes that were integrated into the system. And so, we're seeing an increase in mycorrhizae, and especially the nitrogen cycling area. It was all really positive. And so, just kind of highlighting the nitrogen cycle, we saw a big uptick in that. And yeah, we tested several other products for Russell's soil, looking at multi-species microbials, and we saw that EnSoil looked to perform the best out of all of them. So we're really happy to see the response that EnSoil had on Russell's soil. And if you have any questions about any of these biomarkers, please let me know. It's just really the soil's ability to, cycle nutrients, and then also the overall community health and structure of those systems.
And one of the things I want to add to that is, these tests do cost us money, and we have a fairly decent sized farm to the point where we can economically do them. And if you have a smaller operation and you would like to see some of this testing data that we've done with EnSoil, instead of having to spend your own money on the testing, I know that Meredith has most of them. I'll be glad to share those with other producers so that you can see what results we're seeing. I mean, everything from soil biology and how it's functioning. We've even, last year, we haven't finished this year, but last year we gave them our yield maps on all of the different, plant work that we did. And if that's something that would help producers have confidence in, trying it out, I'm more than happy to share that with other people. And EnSoil kind of took that one step farther. I had never even heard of Dr. James White before. And Meredith kind of made the connection, if I remember correctly. And last year they did a really neat study. When we talk about drought resistance and how much, plant root that we're putting on this plant and accessing, more of the profile, it was really cool to see in the study that the algae was actually stimulating the soil biology, one, and then also it was going inside of the plant root. I kind of nerded out on those videos. Yes, like we can actually see the EnSoil algae and the bacteria, the fungi, everything working inside of the plant root, and the plant root will strip the nutrients off of the biology that was just stimulated by the EnSoil, and then it spit them out, and then it's actually making PGRs, plant growth regulators, inside of the root structure of the plant, which actually stimulated the roots of the plants. And so, that may be one of the ways that we're accessing more profile, which gives us more nutrients, more water, and helps. This year was not a pristine year. We had really, really wet spells and then really, really dry spells. And our plants really didn't seem to suffer where we were using the EnSoil algae. Now on corn, producers in the north that are going to have to dry their corn down, we did see enough plant health, even with the quicker growth in our corn plants, that we were just a little bit higher on moisture content. Having a little bit more moisture content, yeah, we had to dry that corn. But where we're using the algae at and we're seeing the plant develop faster and have a quicker dry down, plus we had more bushels. I would say in the neighborhood, roughly 8 to 13 bushels was a really good average for the last 2 years. And for what we're paying for the EnSoil, to see 8 to 13 bushel differences, plus that drying content, getting that plant through the season and healthier without having those delayed timings, really economically worked out for us. I mean, you're probably talking in the neighborhood last year of about $125 to $130 an acre in increased yield and drying costs. This year we're, corn market's down a little bit, but we're still going to be somewhere in that $80 an acre, maybe 90. And I know that Tucker and Meredith have talked about, 3 or 4 different applications of the product, and we actually were able to do 4 passes this year in our plots, and then we actually had a little bit left over, and I talked to them about it. We just wanted to know what would happen if we did 9, so we actually applied it 9 times. We'll see in the next maybe 2 days what that's going to turn out to be like. We'll be picking that and just seeing how much of a difference can really bumping the algae up. We haven't done the reduced fertility yet. I know that there's some studies that they've done. Are those studies public yet? Yes.
Yeah, we just started publishing some stuff about improved ROI by reducing NPK.
Is the Clemson study out yet from last year?
One? Not sure if it's finished yet.
Well, do you care if I talk about it a little bit?
Go for it, Russ.
Okay. So the Clemson data that they've been working with, they had, 25, 50, and 75% reductions in fertility applied where they were using the EnSoil, and then they had check ships where they were using full fertility. And I know Tucker and Meredith have talked to producers about cutting back on their fertility. We haven't done it yet. 2024 is going to be the year. I promised them we're going to do it. We're definitely going to do it. We're going to do some 25, 50, 75, and I'm even going to go crazy, and we're going to do some 94 strips with no applied fertilizer, just EnSoil Algae, so no pressure there. And we're going to see what it does. But, where we were seeing reductions of applied fertilizer in those studies with the university, they were seeing actually higher yields than, where we were putting all this fertilizer in. And I think it goes back to what we were talking about earlier. When universities came up with fertilizer recommendations 40, 60, 80 years ago, it actually is not based on yield. It's based on yield response to fertilizer applied. And so the higher you go with fertilizer application, the smaller the response is. And the only people who benefited from that was the fertilizer company. The farmers have not benefited from that in probably the last 50 or 60 years, but yet, because that's how we educate our students in universities, and that's how agronomy and CCAs are taught, and how they have to follow that reductionist scientific studying, it really doesn't look at how nature is working. And so we've even played around with EnSoil in our foliars. Instead of using a surfactant, one of the cool things we found out is the algae has a micron size of 5 or less. And so we can actually use it in our foliar sprays and actually see a benefit there where we can actually reduce the amount of liquid fertilizer that we're using in our foliar passes because we see an enhanced efficiency. We've got 2 years of study data that we've been doing these foliar trials and cutting our rates back 25, 30, even 50%. And we have seen a benefit there. And those are some of the, most costly nutrients that we use. So I don't know. Let's see, we're about 7:30. I mean, do y'all want to take a little bit of questions and answers?
Well, yeah, I can kind of segue into one of the questions we got asked. You've talked a lot about the positive effects, and the only negative thing really being how much more growth there is. So can you tell everyone, how hard is it to apply the algae?
It's as easy as pouring a cup of water. I mean, applying the algae, say if we're using it in our in-furrow system on our corn planter. I mean, essentially we just take it, and our planter's roughly doing about 40 acres on a tank, and we measure out, 40 acres times 8, whatever that ounce number comes up to. We pour that in there. That's as easy as it gets. So it's not difficult at all to use it. If it spills on you, you're not going to have any harm, compared to, what we see with chemicals. And so I don't have any drawbacks at all when it comes to mixing it.
Thanks for answering that one.
Yes, I'm gonna stop sharing so we can all see each other for now.
Yeah.
And feel free to put questions in the chat, or unmute yourself and ask your questions to us or to Russell. And Meredith and Tucker are also on the call too if you have questions for them.
I have a question for Russell.
Yeah.
So on your first slide, Russell, you have a picture on the left as I'm looking at my screen, and you say that this is untreated with EnSoil. What is that? What was that soil treated with? Was that chemicals? Was it what another kind of fertilizer? What did grow the corn there?
You're
Is
muted,
that a really
Russell.
tough question?
Russell, you're muted.
Oh,
Thank you. I was speaking to nobody. We did not change up anything except for the EnSoil in our plots. So that farm had 2 gallons of 318-18. It had a quart of fulvic. It had a pint of humic. It had a 2x2 system, which we run. It depends on what the fertility is of the farm. It could have been KPS, which is potassium thiosulfate, or 1137O, or 32%. And then we haven't done a lot of dry fertilizer over the last several years. We've predominantly been with liquid. And so, if we needed additional nitrogen, which that farm did, it got 32% Y dropped into it. And let's see what else that farm got. I mean, it was a full program. So the only difference between where you see the picture on the left and where you see the picture on the right was 16 ounces. At that point in time, it was 8 ounces, but for the full season, it would have been 16 ounces of EnSoil. Because it would have been probably 3 or 4 weeks after that picture was taken that the drone came out, and at Tassel, we put on another 8 ounces of EnSoil.
so in other words, the soils were exactly the same, except the one on the right then received a dose of EnSoil, and that was the only difference.
Yes,
Wow.
that is correct.
It's quite a difference. Okay, interesting.
Thank you. We have 2 questions in the chat just to briefly answer. Somebody asked how often can you apply EnSoil and still get a return? So we recommend that you apply it 3 to 4 times a growing season. That's the company recommendation that is based off of research. Now, we also know that applying it more than that it's not going to harm anything. In fact, it will likely benefit. And applying less than that is also benefiting, just at a different rate. So we don't necessarily know right now the specific data per application, but we know that typically around 3 to 4 applications is where we get the most benefit for both the financial return and for the return on the crops and the soil health. And then somebody asked, can you apply EnSoil without a cover crop in the fall? Definitely, you can apply it to absolutely anything that is growing in soil at any time of the year. Excuse me, I had a sneeze. So you can do this in your house, inside, greenhouse, outside, in the ground, container growing. We have folks using this in their houseplants and on thousands of acres in the middle of the country. So it's really anything. We encourage you to use cover crops, and we'd love to chat about that if that's something you're interested in. Other questions folks have for Russell or for us?
I have another question.
Go ahead.
In terms of pests like insects, worms, fungal things, whatever, does EnSoil help with these, or do you have to continue using chemicals to deal with those things?
Yeah, I can talk about that a little bit, and then Russell, you can share your experience. So we believe that the EnSoil is boosting the immune system of the plant, similar to if a human were to take minerals and supplements that would boost their immune system to prevent illness. So if a plant is at risk of, say, being hurt by a pest or a fungus, their using the algae is going to help support that plant to be more resilient and to adapt to stress better. We don't have specific data currently on like specific pest management or fungus management, but that is what we have found observationally. Russell, do you have any experience with that or something you want to add?
Yeah, I've never tested to see where the Brix level is changed by the algae, but I would imagine having a healthier plant would be less disease.
Thanks.
Okay, we have Eli has a question. So, Russell, you mentioned you do a lot of trials and research with different products on your crops. And Eli's curious, how do you vet the products before using them? Are you ever nervous about them not having great returns or having negative impacts?
I'm sorry, my phone broke up on that. Could you say that again, please?
Yeah. So you do a lot of trials and research with different products on your crops. And just out of curiosity, how do you vet the products before using them? Are you ever nervous about them not having great returns or negative impacts?
Well, until this year, we pretty much just had to take the negative impact. It used to be somebody had a product that we would want to try, and we would essentially give it a shot and put it on our acres. And there are products out there that we've used before, and we've had yield loss. It actually had a negative effect. One of the ways that we're able to combat that now is with Rise Bio. You heard Josh speak earlier. We actually send him our seed, our soil, and we could do a greenhouse study like you saw in the picture and see what the at least biological response is going to be. And then once we know what the biological response is, if it's a positive, then we'll start using them. But we'll start vetting them now using that testing method. We've already got a few lined up for this fall.
Jean had a question in the chat, which I'll read out. I've got a wet, heavy soil prone to compaction, been working with cover crops, no till, strip till in 2016. Does EnSoil do much? The number of applications Russell has is pretty intimidating money-wise. Can you make much difference with 1 or 2 applications? I'd like to start doing more plant tissue testing during the season. This is the landowner, not the farmer. Great question. Thank you, Jean. Yes, EnSoil does do a lot, and there are a bunch of aspects to this question that I want to address. So wet, heavy soil and soil that's prone to compaction can be impacted in a lot of different ways with regenerative strategies, and EnSoil is one of those, but specifically growing crops that are going to have roots that break up that compaction, and also microbial activity to break up that compaction, the mycorrhizae, the different microbes that are in the soil that are going to help strengthen that soil and allow water to infiltrate. These are all things that are going to eventually help a wet, heavy soil. So with EnSoil would fit well into that system. And I can see that you are asking about the number of applications and the cost. So we have not mentioned this on this call yet, but this is a great time to do so. So our pricing for 2024 is going to look a little bit different than how it did for 2023. We have a pricing matrix that will allow growers to purchase EnSoil and use it appropriately for their scale of agricultural use. So folks that are growing on specialty crops or a small amount of acreage will be able to use it at closer to $20 an acre, and folks that are growing large scale crops on lots of acreage will be able to qualify for a reduced price closer to $9 an acre. So we can talk to you more about the specifics of that pricing if you are interested in the calculations. We just released something in our weekly seed today that explains a little bit of that. But that being said, we do recommend 3 applications a season at least. You will have benefit. The algae will be doing something with fewer applications. We just recommend that 3 is the best case scenario. And if you are to order, if you are to purchase the algae and place your order for 2024, we are offering incentives to do so, where you buy 2 applications and the 3rd application is free. So there's a 33% reduction on that. So I hope that that answers your question, Jean. I'm happy to talk about it a little bit more, and I definitely encourage you to do plant tissue testing. It's great to be able to see the data with your own eyes, the observation, but also those numbers of your analyses.
And I just wanted to say that on average, we're applying 2 applications. So on corn, we have seen the highest return with 1 8-ounce shot in furrow, and then we're coming back with another 8 ounces foliar over the top of corn. And then on beans, we just haven't seen beans respond as much in furrow. I mean, we have had pretty big results, but they haven't been consistent in furrow. I think a lot of it has to do with the shape of the ground, but 2 shots over beans foliar. So we're making a V3, V4 pass on beans, and then we're coming back at a R1, and we're doing another 8 ounces at R1. And we've seen a pretty big return with that. And that's just 2 applications. And then we've got our plots that, yeah, we're pushing them to see, is there a higher threshold to that return?
Thanks, Russell. Travis asked, have you tried using the EnSoil as a biological primer on the seed as a seed treatment? So this is sort of what Russell has been talking about with in-furrow, applying the algae at seeding. And actually the research that Rise Bio did, that graph that we showed you comparing Russell's seed and soil in a greenhouse versus in a greenhouse untreated, that was at seed. So that EnSoil was applied in furrow at seed with Russell's soil, and then there was a control and there was his test, and those were in the greenhouse. So we are really interested in looking at seed treatment, and this is something that we're putting energy and research into currently, because we do believe that we can see similar benefits, as you saw in that graph, with using the EnSoil on the seed.
And I'll just say that's something that me and Tucker have talked extensively about. I think if as a farmer you have the ability to treat your own seed and apply it within a short window, then a seed treatment would work great. But if you look at how our commodity system is currently set up, if you were going to try to send EnSoil to your seed dealer and have them treat it on the seed, and then you're talking 120 days, 90 days until you're going to put that seed on the ground. I mean, that's the one thing that I think most people don't think about with these biologicals. It's a living, functioning organism. And so I know that they're doing some testing to check the viability on that. If that happens, that would be great. I just get it thrown on my seed. I just think we have to figure out, what that time window is from treatment till planting, so we don't see any degradation in the product.
Great, thanks, Russell. I see a question in the chat about the graph, so I'm going to put the graph back up for everyone to see. And then, Josh, are you still with us on the call? Are you able to address this question about the untreated versus treated bars?
Yes. Yep.
Great, thank you so much.
So I saw the question there, and it's talking about the nutrient cycling processes. One of the things that is not in this graph is the organic nitrogen breakdown of nitrogen. So specifically looking at nitrogen fixation, the treated or the untreated section is higher. This is obviously a factor of Russell having very healthy soil. But another thing that isn't noted in here is the urease gene, which is one of the genes associated with providing organic nitrogen directly to the plant. And so that was the nitrogen cycling potential could be, or the nitrogen could be getting found directly in the soil from organic nitrogen breakdown. So that's something that maybe isn't represented here. And then as it relates to some of these other metrics, I would say, the biggest thing in the early forming rhizosphere is which is what we're looking at, that initial first week, is we want to see the community structure form properly. And so once in the early stages of the plant growth, that we're seeing a higher observed species and more mycorrhizae present. As the plant matures, those other factors or those other nutrient cycling potentials are increased. And so I do want to say, it may look like some of these are lower, but specifically for denitrification, that's a good thing. And then also for nitrogen fixation, the plant is finding other means of getting nitrogen to the plant. And so maybe it's a function of our technology. Absolutely don't think it's a factor of the EnSoil.
Thanks, Josh.
Question. Yeah.
Yazzie, did you want to share the questions that were sent before the webinar?
Yes. We've got those.
And feel free to continue putting questions in the chat. If there's anything that we don't get to, we will send you a personal message with an answer to your question or give you a call, because we'd love to chat about the things you're curious about.
I just wanted to say real quick that I appreciate everybody dealing with any kind of noise you had here for the first little bit of this. It's been a long day of farming. I'm sure everybody understands that. But I'm finally done for the day. I get to go home and then work more.
We appreciate you, Russell, and your time.
And I don't mind staying on just a little bit longer if y'all have them, if y'all want to answer questions if we have them.
Well, yeah, let's see what we get through. We'll probably stop at 8, and then if people have more questions, we can direct them to you over email or something. So some questions we got before. One of the questions from Linda was, corn often requires extra nutrients. How often do you use your EnSoil algae?
On average, twice. On some of our degraded ground, we've done the 3 shots where we've had 3 different application windows. But most of our farm ground that we've got now, we've been farming with regenerative practices for 8+ years, and so we're already seeing pretty good cycling, and we're using this as an enhancement twice on average.
Thank you. And then we have a little cluster of questions. First, do you know of other farmers near you that are testing and using EnSoil?
I would say near is a relative term. Yes, there are other farmers in my side of the state that are using it. Some of them are not using it in the same manner I am. They're using it on pasture. Some are on row crops. Some are on vegetables. And I haven't heard any negative feedback from them as far as their forage quality, grain quality. It seems to be working as it should be. We've only had one producer that's really ever said that it didn't really give them the best results. And, after we started talking to them, the way it was supposed to be applied, their volume on their sprayer was actually too low. So when we're doing foliars on young plants, we're running about 15 gallons. And when we start getting into, V8, V9 on corn, you've got a pretty good amount of biomass there. We'll actually bump our rate up to about 20, 25 gallons. And, they were in a situation where they didn't want to have to haul water. So they were running it at about 8 to 10 gallons an acre. And I think it just came down to a coverage issue. They didn't get adequate coverage, and so they didn't see results. They saw results on the first part of the season, but they didn't see results when they had to bump back on application rate.
Okay, thanks for sharing that. Okay, and then can you just talk briefly about your data for this year? For example, what was your yield this year and what did you do differently?
So they haven't released our corn yield data, and we just picked our beans today. So, you know, the ones that the crops that we're pushing to the extreme, we don't have a lot of data on those yet this year. But our normal production corn, the last farm we picked was, 192 to 210 bushels. Our county average is 100 to 110. So, I would say that we're seeing a huge benefit to giving these microbes an enhancement in the beginning of the season. You were asking about insects and disease earlier. We didn't apply any fungicide this year except for one farm, and that was more of our fault with variety selection. And we actually didn't spray any insecticide this year. And so, I think it's one of those things where if we can keep the plant healthy and keep it fed and keep the microbes fed, we just don't have to rely on a lot of the other inputs. But if it came down between losing a crop and using one of those inputs, we're gonna have a crop to raise.
Yeah, that makes sense. Another question for you, Russell. I know you mentioned this briefly to me and Yazzie, but would you talk a little bit about your fall trial for decomposing your residue after harvest?
Yeah. So, you take 150 to 200 bushel corn, there's a massive amount of fertility that's tied up in that residue that we didn't harvest off of the field with the combine in the grain. And so one of the things that we're looking at is there's a bunch of companies that they sell bacteria and other microbes that help decompose residue, and we're trying some of those. I always tell Meredith I always like to put products up against products in the same field. So this year, as we're getting through corn harvest, I think the new term is called biological tillage. And, farmers are looking at ways to increase microbial efficiency in the fall to break down residue, which can help with disease and pest cycles. It can help with nutrient cycling. And so we've picked out 3 farms this year. We're not really going to focus on beans because there's just not a lot of residue with soybeans. But we've got fields this year that we're going to split apply, and we'll put the EnSoil Algae out. I think we're going to try a 4, an 8, and a 12-ounce rate, and we'll actually take 1 square foot measurements in random spots in the field and flag them and weigh what the residue is right after harvest, and then we'll apply this EnSoil, and then we'll look at it probably 15, 30, 60, and 90 days and actually weigh out and see, how many grams of residue matter have we helped to decompose and not have a breeding site for disease and insects that are in the winter time, and also see those nutrients re-released back into the profile, which are going to help feed our cover crops and also our subsequent crop the next year.
Awesome. That's going to be super interesting to hear the data back on that. And Travis wanted to ask, would adding molasses with the EnSoil help to break down residue?
Yeah, I think adding a sugar source to that would probably definitely be a benefit. Just enhance the efficiency of what the microbes are trying to do. We haven't ran molasses because we haven't been able to find a good a good grade of molasses that we can see consistency out of. So we use sugar in furrow, and we use sugar over the top with the EnSoil already. And we can buy granulated sugar cheap and in a lot of places. But yeah, molasses or any type of, sugar source, I think would also be a benefit to that. But I don't think that we're going to do that, because once again, looking at it from a research standpoint, we're just trying to see what we can do. So I think if we add molasses to it in our plots, or if we might break down and add some and do like a 90-foot strip with added molasses, but I'm just kind of interested to see, what we're going to get straight from the algae, but we might mix a little bit in and see if there's an enhancement there as well, like we've seen on the foliar sprays.
Great, thanks, Russell. I'm going to our contact information, and feel free to ask any more questions as we start to wrap up.
Yeah, this is Randy. Can you hear me?
Yeah, hey, Randy.
Hello. Hey, Russell, you were talking about trying to get the soybeans out of the combine or into the combine. What was the problem there? Or
I'll share some pictures and I'll let EnSoil post them on their page. But the problem is these soybeans some of them are realistically the size of a pinto bean. Our seed size is absolutely phenomenal this year with the program we've been using, and our seeds per pound have been coming in at 16, 17, 1800 seeds per pound. Industry standards probably going to be around that 3,000. So our seeds have been about double the size, and just where the triangle pieces are in the grain tank, we run those pretty tight. We don't run them wide open, and tomorrow morning, I guarantee you we'll be opening those up quite a bit, just because we've been fighting this for a little while. But we're going to try to get out some of those, like I said, those soybeans that they look like a light brown pinto bean. We're going to get at least a couple handfuls of those out of the hopper in the morning before we haul to the elevator, and we're going to do some seeds per pound, because I really think that those soybean seeds are actually going to come in under 1,500 seeds per pound, and that's one of the biggest yield factors for soybeans that a lot of producers don't think about. Is if you had 120,000 plants out there and you didn't add any more seed, you just went from say a 3,000 down to a 2,000, you're probably adding north of 25 bushels, just in seed size alone. And so, like I said, we haven't had a lot of years like this, but as we continue down the program and we're starting to get our timings and everything figured out, through the testing and when to apply these products, we're starting to see more and more gains. So, last year the farm averaged about an 82 bushel bean average across all the acres. This year, I'm hoping we're going to be somewhere north of 90, the way it's turning out.
What's your test weight on your corn?
This year on corn, what we've harvested so far, we've been averaging anywhere between 61 to 64 pounds. If I had to pick an average, I'd say we're probably around that 62 pound test weight average. And that helps a bunch with the products that we're making. I mean, most of our corn is going to food or alcohol production. And so, definitely a lot more nutritional denseness in those kernels than what you would see on an average farm, I would say, in the area.
Is that the inbred corn? What is it?
The open pollinated?
Open pollinated, I'm sorry, yes.
Yeah, so we do grow a hybrid non-GMO. We grow, I would say about 20 to 25% of our acres are actually traded varieties that go to a feed mill, that we sell a little bit of our extra corn to a feed mill. We grow traded corn on those, and then we grow quite a bit of open pollinated. The open pollinated, we've only harvested the Jimmy Red and the Bloody Butcher. We haven't got to the Reedsy Aladent or the Blue Claridge, but the two red corns so far have been in that 59 to 60. I think we had one of our tests came back about a 61, and most of that was averaging 18 to 20% moisture. So as we dry those down, in our grain bins and get them down to moisture weight, I would say those test weight numbers would increase as we dry that grain down.
Excellent.
Thank you so much, everybody, and big thank you to Russell. We appreciate your time. Please feel free to call any of us, reach out to us via email. You will all be receiving a follow-up from the webinar with the recording and with some additional information to add to this conversation. So we really appreciate your participation, and if you have any more questions, feel free to stay on and chat with us.
Thank you, guys.
Thanks for having me.
Thanks, Russ. Appreciate you.
Thanks a lot, guys. Have a good night.
Thanks.
I was wondering on the soybeans, the corn was matured faster, if the soybeans matured faster too. I didn't get that.
We have not really dove into the soybean maturity stuff yet. This year it's honestly been a weird year, and it's not just been my farm. So we planted and then we flooded within like 12 hours. Then we replanted, and then we didn't get rain for like 45 days. And then we had another flood in June, and then we didn't get rain for like almost 50 days. Then we had 2 tornadoes in the same day. One of them actually went through my farm. Our determinate beans have actually finished before our indeterminate beans, and they were planted 3 weeks later. So as far as telling maturity, as far as timing goes with EnSoil, I haven't been able to nail that down with soybeans. Maybe that's something we need to pay a little bit more attention to next year.
Did you think, did I understand it right, that maybe corn might mature almost 10, 12 days earlier than normal?
Yeah. If you wanted to look at it, I mean, essentially, we're probably losing a 1/4% of moisture down here a day. And so, like, if you go off of that rationale, then, at 2.1, you're looking roughly 9 days. 9 days different in maturity. So, and that would be about right, even with the leaf stages that we saw all season. I mean, we were roughly 1.5 to 2 leaves ahead all season long. And so either by moisture count or by leaf count, it was steadily about 8 to 10 days that we were seeing that plant able to develop, because it just had, it was healthier the whole time. We didn't have a slowdown during the dry times, which we typically get.
Now, would you, have you done anything, put EnSoil on your cover crops at all in the fall?
We did last year. We didn't have a lot left over, so we only did probably 10 to 15 acres in one field. We were able to see about, in the neighborhood, maybe a 5%, 10% difference in biomass. We were so far behind this spring. A lot of people may not know this. Last fall, during the middle of harvest, Heather fell and completely shattered her L1 vertebrae, so she was bedridden for about 2 months. And so there's a lot of the testing that we just didn't get to perform that we wanted to, but we did see a biomass difference.
I can't wait to listen to you at the Iowa Soil Health Conference this year.
Yeah, we'll, by the time we get to the big soil health event, we'll have a lot of our plot work and our plot data finished up with EnSoil and all the other products we tried this year. And we're going to try to talk a lot about return on investment and what we were able to see on the farm and what worked and what paid.
Wonderful. Absolutely wonderful.
Thanks for your questions. And thank you
You
so much
bet.
for helping us out with the RISE biograph interpretation. We appreciate you.
Yeah, absolutely. Thank y'all.
We hope to have a webinar in the future with Josh or a member of his team to talk more about the biology at his level in the lab and in the greenhouse.
Yeah, and we only did one. That was only one sample, so we're only looking at that one greenhouse sample. And so, with metagenomics, you're looking at billions of microbes, and so trying to make sense of it all is not an easy task, but we do our best. The more data, the more samples that we can run, the better, the more reliable information that we can provide.
So I was just able to look here at all the Yazzies that we have in here.
Yeah, I'm so sorry to anyone that I removed at first. I genuinely was terrified. I thought someone's hacked me. But yeah, everyone is mean tonight, and it's
Russell,
interesting.
I had a quick different question for you. Is the storage of EnSoil an issue at all? Having to refrigerate it or not?
That's a great question. We recommend that it is refrigerated or kept in a cool, dark place. Mainly, we just don't want to keep it in direct sunlight or in heat. Those things will decrease the shelf life of the algae, but it's meant so that we've been able to keep the algae cells alive in the dark for many months. And so with refrigeration or with storing it in a cool, dark place, that is able to happen.
Well, Camille, I know you guys have. That's why I was asking Russell. The real world is sometimes a little different.
Sure.
I mean, last year when we didn't have a ton of it, I just had a refrigerator up in my office, and we just kept it in the refrigerator. This year, I kept it either in my garage of my house, which it's not perfectly climate controlled, but it stays typically below 70 degrees, or I just shoved all the jugs in my basement. It stays a lot cooler down there. And, when I needed them, I just grab them out. And, this year we did not have to refrigerate it. And we've still seen, the testing with like what Josh did and the PLFAs and the Haneys that we did. We didn't see any difference than what we expected on the data.
Okay, that's what I had heard too. Interesting.
Yeah.
Makes it easier. Well, thanks again.
Yeah. Well, like I said, if anybody's got any follow-up questions, you can email me or call me, as long as you don't mind listening to equipment during harvest, or I'll see you at the big soil health event.
Thank you all so much.
Thank you,
Oh,
guys.
Thank you.
See you soon.
See y'all later.
Thank you.